Perfect. Okay, hello everyone. And I'm really excited to be hosting this meeting today, and thank to the speakers to accept the invitations. Just some quick notes. During the whole session, everyone is going to be muted, and please stay so throughout the whole session. Also, if you have any questions from the speakers, please write it in the chat section. If you have enough time, we'll make you unmuted to be asking your question directly. Otherwise, I would relay the questions to the speaker. So without further ado, let's go to our first speaker. Lukas, you can go ahead and share your screen with us.
Perfect. Thank you.
Sure.
Thank you, Zeinab and Forsan, for hosting and co-hosting this session. Okay, so my name is, everybody can hear me and can see this well, probably?
Yes, you're
perfect. So my name is Lukas Hirsch. I'm a graduate student from the Neural Engineering Lab at City College, New York. This title is using machine learning for robust brain segmentation for TES modeling. So for those of you who were on Monday on the very nice talk of Andy Huang, where he talked about ROAST, a realistic volumetric approach to simulate transcranial electric simulation. This is a tool that does modeling for current flow simulation in the brain. So this is the pipeline, the main pipeline of ROAST, and I'm going to be focusing on the first step, which is the segmentation step. In this step, taking as an input a subject's MRI, a segmentation process happens, which is the process of assigning each pixel or voxel in the image to a specific tissue label. In this case, because of the relevant electric conductivity and electric properties, we do segmentation of 6 tissue types, which are gray matter, white matter, cerebrospinal fluid, or CSF, and bone, sinus, air, skin, and then background. So there are a lot of automatic segmentation tools like FSL and in this case SPM from MATLAB that work well on healthy subjects. But on a clinical context, we've been having a lot of issues when finding abnormalities in the brain, either through lesions, tumors, or different abnormalities that you can find. So most of these automatic segmentation tools work using an atlas or prior information on tissue location to help do the segmentation. And these models are often not flexible enough to account for deviations from normal anatomies. So in this example, in the middle, we have a T1 weighted MRI. And in the middle, we see a stroke lesion that after suffering necrosis, the tissue is filled with cerebrospinal fluid, leaving this dark mark in the middle. On the left, we have what we call our ground truth segmentations. That are segmentations manually made, which is how we usually do corrections for the segmentations that we normally have. And on the right, we see an example segmentation made by SPM, which is currently in the pipeline of ROAST. And this is the type of errors that we usually see, where ROAST SPM does not expect to see CSF in this location, and it mistakes this with the expected white matter. And this type of error then propagates to the rest of the brain because it learns the wrong intensity distributions per tissue that it's classifying. So this is another example. In this case, we have an abnormal anatomy. And this is not because of stroke. This is a very enlarged ventricle. And again, here on the left, we see the ground truth manually segmented. And on the right, we see the normal segmentation result from SPM, where it expects a normal size ventricle in the middle, and then the rest, it assumes that it's gray matter, learning again the wrong intensities for the tissues, making errors that propagates through the whole rest of the brain. And so our approach would be to try to replace this segmentation step with state-of-art deep learning tools. So in natural image processing, there's been, of course, the last 10 years, a huge breakthrough using convolutional neural networks for image recognition and image segmentation. And this is also transitioned to biomedical image processing. Now, the use of atlases and priors hasn't been picked up by the machine learning community as much because of the usual trend of trying to make multi-purpose models that take the least amount of regularizing or constraining information possible. In this case, for these task-specific tools, because there are a lot of standards that follow biomedical imaging, there's a lot of useful prior information. This is why also SPM uses this. But we would rather like to use a deep neural network, in this case, a convolutional neural network, to decide on its own how much this prior information should weigh in on the final result. So this is a very basic three-dimensional convolutional neural network. This is going to be the backbone of the models that we're using. So this takes as an input an MRI, and the specific input is a small high-resolution volumetric patch from within the MRI. And after processing through several convolutional layers, the output would be the prediction for the class label of the center voxel of this patch, what we call. So after processing for all non-overlapping patches, we get the final segmentation of the whole image. So to this, we can add different modules that are independent from this initial basic model that we call the detail model. So secondly, which is also common to increase surrounding information, we add what we call a context model. So in this case, we take a larger, low resolution surrounding patch to alleviate computing resources. So this adds extra surrounding or context information to this. Now at this point, this architecture is analog or similar to another existing architecture called the DeepMedic, which is also a very successful biomedical segmentation tool. We also add another layer, which is we call a tissue probability map, giving spatial priors layer. So this is an independent layer that includes information specific per voxel in the image. And this is a tissue probability map. As the name says, it gives a value, a probability value per specific absolute location on that voxel belonging to each of the 6 different classes that we're trying to classify. This is the same tissue probability map also used by ROSE at the moment. And finally, we can add some prior information on what we know about brain morphology. This is doing post-processing the finished segmentation by the model. This is applying a conditional random field. This is a structural probabilistic model. So this is a model that assigns different probabilities to different structures. In this case, one structure is a finished segmentation. And we can add our own knowledge on brain morphology by penalizing neighboring labels. For example, if we see that the output of the model gave out a skin label right next to CSF label, because we know that's not expected in normal morphology and anatomy, we penalize those configurations. So this further cleans up the final step. So adding those 3 models together gives this model on the right. So this is a basic detail model, adding context, adding the tissue probability maps, and adding the conditional random fields. So this is context aware, this has absolute location awareness, and also morphology. So we call this a multiprior. So first, just to give an example of how the basic detailed convolutional neural network performs against SPM. So in this case, not only we have an example where there's a lesion inside that SPM here on the left cannot recognize, but also we see, for example, the unflexibility of this model. The neck position of this head is a bit unstandard, and SPM fails to recognize the positioning of the head. Now, these are problems that a basic convolutional neural network can easily solve, detecting all boundaries in the image. But we see it makes some qualitative errors. So here in red, it's a class label for skin tissue. And we see it confuses brain tissue with non-brain tissue, placing brain tissue outside the brain and non-brain tissue within the brain. So for this, adding the 3 other models and the multipriors, we see that all these problems are resolved, giving a clean segmentation. Now, progressively adding all these 3 modules in different combinations, we see that each one of them provides a gain as measured in dice score. Dice score is a measure score that ranges between 0 and 1. 0 being the worst agreement against the ground truth segmentation that we're comparing against, and 1 being perfect agreement with the ground truth segmentation that we have. So in this case, on the far right, we have the multiplier with all modules added, which averages around 0.88 on dice score. This is on 42 size test set of stroke MRI and 4 healthy subjects. We see from all these 3 models, the one that provides the most gain is adding the TPM, as the 4 models here, the best performing models had. Also, when seeing the specific tissue performance, so taking all of the 6 tissue types that we're classifying, when comparing a model that doesn't have the TPM, so similar to the dipmetic, and a model just adding the TPM, we see an increase in all tissue types. This is a visual example of the type of errors that adding a TPM corrects. So, for example, these are mainly by not confusing brain tissue within the brain cavity with non-brain tissue outside the cavity. So these are, on the left, there's the ground truth. And here we see the model without the TPM. We see this is introducing background within the brain on these dark regions that just look like background. And the same here, this in the neck tissue looks a bit convoluted, and sometimes it mistakes with brain tissue. But adding the TPM solves all these issues. Also, we compared this using other current automatic tools like FSL, SPM, and QuickNut, which is another convolutional neural network for doing tissue segmentation. We see this is all different tissue types. We see on healthy tissue types where there are no deviations. Gray matter, white matter, bone and skin performance is usually good by SPM, for example. But wherever there are lesions that are filled with CSF, these models fail to recognize. So then for a validation with an out-of-sample data set, we had a set of MRIs from a different study. These are disorder of consciousness subjects, and they're characteristic because they have very abnormal anatomies. These are subjects that usually SPM and other automatic segmentations to fail considerably to do a normal segmentation. So this is an example, again, of a result of the multipliers on this kind of subjects by achieving nice recognition of the different abnormal structures. Also compared with the other tools, they seem to perform better. Of this data set, SPM and FSL and QuickNut, they seem to be a lot worse on finding the lesions or the abnormal ventricle enlargement in this data set. FSL in this case seems to be a bit better. So for this, we manually segmented 10 heads from scratch and used that as a ground truth. And so to summarize, we've shown we can replace automatic segmentation tools with deep learning models and considerably reduce the anatomical errors, which are relevant for current flow modeling, as missing a huge chunk of CSF within the brain will affect modeling of the current flow considerably. And with biomedical data, I can show that leveraging prior information also generates better segmentation results, especially in these cases where we have small training samples. Oh, thank you very much. Are there any questions?
Wonderful. Thank you very much, Lukas. And if anyone has any questions, please ask in the chat. And I have just one quick question.
Yes.
This machine learning models your plan is going to be added to the ROAST, or?
Exactly. So
it's a new generation of the ROAST models.
Right. So the plan for the future is replace SPM with this model. So overall, the long-term plan for ROAST is to make it more independent from MATLAB. And this would be one of the steps, SPM being a MATLAB tool, and at the same time correcting all these anatomical inaccuracies when working with clinical subjects.
Another question, how do you, I guess Fang is asking the question, how do you calculate a tissue probability map?
The tissue probability map was made with an average of a couple of hundred subjects. This is the same tissue probability map that is currently being used on ROSE. So the ROSE paper, there must be a citation. But yeah, it's just an average from healthy subjects. I think around 200 subjects that were used to average the specific tissue location for these 6 specific tissues. So in essence, you could make your own tissue probability map for any of your specific task that you're doing.
Mm-hmm. Great. And I guess Maria Clara is asking some comments on how easy to use it.
How easy?
I don't know what does it mean by it.
This model can be also used independently of ROSE, of course. So we trained this model on this set of stroke patients, and it's uploaded on the Paralab website.
Okay, so it's already available online.
So it is available online. Yes, so in essence, you could download this model. There's still a lot of dependencies on Python that you would have to consider, and being a deep learning tool, it has some dependencies on GPU usage that we're trying to make easier to use. But yeah, this you can download the models and apply them independently from ROSE.
Okay, perfect. Thank you, Lucas. So I'm going to be clapping on behalf of everyone.
Yes, thank you.
And our next speaker would be Dr. Rashed from Nagoya Institute of Technology in Japan. Dr. Rashed, we are really excited to have you here. You can go ahead and share your screen with us.
Thank you. Thank you. Okay, can you see my slides now?
Yes, we can see it. Yes.
Okay, so good morning, good evening, everyone. So my name is Essam A.Rashed. I'm from Nagoya Institute of Technology. And today, actually, I will talk on a topic which is very similar to the first speaker. So Luca had made it quite easy for me to explain what we are doing here. So the target of this work is to try to find the semantic segmentations that is mainly for personalized deep brain structures for non-invasive brain stimulation. I think most of you are now aware of why we need the segmentation, or why we would like to make a human models to be used for the electromagnetic dosimetry application or brain stimulation. So the common clinical application that we are focusing here is brain stimulation, something like TMS or tDCS. And the main issue which we are aiming at the long run is to find some way to create a personalized head model in a way that can be operated at the clinical scale or the clinical level, which means that we would like to acquire the medical images, and then we would like to have some tool that can create head models in a very short time, which means that we would like to have a very nice segmentation and also in a very short time as much as possible. So to go through this, we actually interested in machine learning based on the recent development of deep learning. Precisely, we find that it leads to great success in many image processing and analysis applications. And here we would like to investigate how to design a deep learning architecture for human head segmentation. As you know, that the pipeline or standard pipeline is actually started with the MRI image, and then we apply some kind of segmentation, manual segmentation or automatic segmentation. After that, we have some kind of head models, and then we have some tissue-based electrical properties, such as a conductivity, to generate what we have, or what we call the volume conductor, which can be used in the electromagnetic stimulations. The problem here is that the segmentations actually is a time-consuming process, and when we focus on the MRI, we find that the non-brain tissue is actually very difficult to be segmented, because the medical scanners are actually tuned so that it will provide the brain tissues in a high contrast other than the non-brain tissues, which is medically of less important. So usually the non-brain tissues are of low contrast or actually appearing in very limited regions. Another problem is that the structural MRI is known for grayscale variations, even with the same subjects. If we scan one patient at the same scanner, different times, we can have different grayscales, as long as also the inter and intra-subject variabilities. So with this large degree of freedoms, we would like to find some way to do segmentation in a short time. So recently we have developed what we call ForkNet, and this is a deep learning architecture for automatic segmentation of the human head models. So I quickly will go through this architecture, which we call ForkNet, and then we will present here what we call sub-ForkNet, which is an extension, especially for the deep brain structures. To explain quickly what is deep learning and why we are interested in the convolution neural network, we can think of this approach as just a mapping between the anatomical images and the segmentation of these images. And then we have here some kind of network. A network means that it is some kind of nonlinear operations with hundreds of millions of parameters that can be optimized to map these two set of variables. So when the network is trained, it can be do the segmentation without actually using any additional priors about the tissues to be segmented. And this is how the deep learning can do the process in a nice way. So we actually interested on what we call convolution neural network, which is actually now the state of the art for image segmentation in many applications, which is simply do some convolution operations around some region in the image with some filter, and then added some bias, and then we estimate how this pixel is likely to be from different set of tissues. For the architecture that includes CNN, it actually operated some other functions, such as the ReLU functions and the pooling functions, which is very simply to be understood. The main approach which we would like to emphasize here is that there are actually a plenty of studies that use the deep learning for brain segmentation. But most of these studies are actually focused only on the brain structure. So something like CSF white matter and gray matter, or even trying to find some abnormality within the brain region. But what we have tried to find is that the non-brain tissues are actually ignored in many studies, and we hardly can find a deep learning structure that take into concern how to segment the non-brain tissues. So this is the architecture which we call the ForkNet. It consists of a set of encoders and decoders. So the blue blocks here, which we call the encoders, and the other branches here, this is the set of decoders. This is a ForkNet of degree 2, which means that the input is the MRI image and the output is two structures. Here we have a sample of binary images representing gray matter and white matter. And this is detailed of each block. It is actually a composite of different layers. I will not go into details about this, but actually you can find details in this reference, and also the open source code is available from this link. So ForkNet actually designed to segment the 13 head major tissues, including skin, fat, muscles, bone, blood vessels, and also the brain structures, and so on. So by using this architecture, we actually succeeded in doing the segmentation for different head tissue up to 13 different tissues. The next step is that we now more concerned about the deep structure, not only the main head structure, but also the deep structures of the brain. So we design also what we call the sub-forknet architecture. In that case, we start with the MRI image with skull strapping. We just keep the brain tissues. And then we have used this architecture, which consists of single input and 7 output. Every output is assigned to different deep brain structure. And then here, it's actually similar to the forknet, but the number of the output are actually different, and the design is a little bit different. So the novel feature here in forknet and sub-forknet from the standard method, which is mainly based on what we call UNet. This is very famous deep learning architecture, is the split of the network decoders. We have found this trick is very interesting and lead to improvement in the quality of the segmentation when we just split the output to each structure independently. So to put it all together, we have designed the following end-to-end architecture. So we start with the MRI image. We segment the main head tissue by using forknet, so we have 13 head tissues. And then we use this information to remove the non-brain tissues. And then we use the sub-forknet to segment the deep brain regions. And finally, we have a head model with 13 main head tissue and 7 deep brain regions. We also do the segmentation over different slicing directions. And we have found that after doing the slicing around different directions, so we used 3 architectures, each for slicing direction. We have found that after doing the aggregation with a voxel-wise majority voting rule, the segmentation is quite improved compared to the segmentation along just one direction. And here we have the data set and training strategy. We have used 18 subjects. We have used T1 structure MRI. And then we have used the ground truth segmentation using the method presented actually in this reference by Laxo et al. And then we use this segmentation as a golden truth of our segmentation. We use a cross entropy loss function that minimized by Adam algorithm. The network is actually, we use 50 epochs with batch size equal 4. And we have found that the forknet requires around 40 minutes for training by using leave one out strategy. This curve represent the behavior of the loss function at every direction. And these are the tissue conductivity we have used for the tDCS experiment. So here are some results for 4 subjects. You can see here the input image, and this is a magnified region MRI. And this is a true segmentation by the semi-automatic method, and this is the segmentation by sub-forknet. You can see here some quite matching results. But actually, we have found a quite difference between the different structures. So for salamus, caudate, and putamen, we can find that the high consistency for the segmentation. But actually, for other structures, such as accumbens, we can find that the segmentation is not so good. So if you look here at this graph, you can find the quality, which is according to the dice coefficient here, along different directions. So we find that the aggregation actually that represent in green color here is actually improved quite much when we compute the aggregation from different segmentation along different directions. And here the segmentations with different network parameters. So we find that when we increase actually the depth of the network, we cannot find improvement of the segmentation. Actually, we find some decrease of the dice coefficient when we improve or when we make the network more depth. Then we have used the segmented model actually for doing some tDCS experiments. So here's the montage we have used. We have used 1 millimeter thick rubber sheet, and this is a conductivity 0.1. The electrode is actually 5 by 5 centimeters with 5 millimeter thickness. And we actually place the electrode around C3FP2, which is close to the motor cortex suborbital position. And here you can look at the tDCS results. So this is a distribution of electric field for one subject at different segmentation scenario. The one who's the left side, this is the segmentation using the semi-automatic method, which we consider as the golden truth segmentation. And here, this is a segmentation where the head tissues is segmented with a semi-automatic and the deep tissues is segmented by sub-forknet. And here on the third column from the left, the head is segmented by the forknet architecture and the deep region is segmented with a semi-automatic. And finally, on the far right column, you can find both the head region and the deep region are segmented by using the forknet and sub-forknet architecture. So mainly, we cannot observe any significant difference between the segmentation by using different strategy. The only difference is the time. So actually, the semi-automatic method is a time-consuming process similar to any automatic method that's actually considered some prior information about the subject and about the combination of the tissues. But here we can see that we can obtain the segmentation actually a few seconds, maybe less than a minute. And here some numbers about the global error and local error at different regions and the average around the deep region and within the whole brain. As you can see here, the global error and the local error are actually within a small values. This is a percentage of the relative value. So it is within the range of accepted values. To conclude, so actually we have here developed the CNN architecture for automatic generation of human head model. So we have segmented 13 basic head tissues and 7 deep regions. The architecture used for the head tissues is called forknet, which is we can say that this is a first deep learning architecture for forehead segmentations, as most of the related approaches are actually segmenting only brain tissues. And we have found that the segmentation accuracy, which is within few seconds, are of high consistency with golden truth, which required a quite long time in terms of dice coefficients. Both architecture, the forknet or sub-forknet, can facilitate the quick generation of personalized human head model. So we hope that lead to a more clinical feasibility so that we can generate a more personalized head model immediately at the clinical level. So we would like to say that the code is freely available at the GitHub repository I have shown before. The tDCS studies we have conducted here is show that the electric field distribution is actually of high consistency. Yeah, finally, I would like to acknowledge the grant from MIC, and I would like to thank my group here, led by Professor Akimas Hirata at the Nagoya Institute of Technology. So, and thank you very much.
Thank you so much, Essam. I'm just clapping on behalf of everyone. It was awesome. Yep. So in the interest of time, I'm going to go to the questions. Peter is asking, how about the ForkNet's performance compared to the SPM12?
Well, so we have not compared the ForkNet actually with SPM. So what we have done so far is to compare the ForkNet with the semi-automatic segmentation. And maybe in future, it's interesting to look at the comparison between ForkNet and SPM. It's quite interesting to do so.
Okay, Feng is asking, you use semi-auto as a reference for validation. How did you do semi-auto?
Again, please.
Feng is asking, you use semi-auto as a reference for validation. How did you do semi-auto?
For the semi-automatic method, this is actually a method developed by Professor Laxo from Aalto University. And this segmentation is semi-automatic, which means that some part is automatic, which is based on free cipher, and some part is actually how to say manual priors is added to the segmentation. So we have used this as a reference, as a true segmentation for our reference, which we have used to train the network, and then we compare how the network is trained these features, and we assuming that the semi-automatic is a golden truth ones. So this is why, because we cannot find any available free data set with the segmentation of several head tissues, including large number of head tissues that's freely available. So we hope in future we can have a more data set with, let's say, manual segmentation that can provide more information or more accurate information to be used for the validation. But so far, we can say that as long as that network is well trained, it can do good performance. So it's actually, it depends on how accurate is available segmentation.
Okay, I'm going to unmute Andy. He has some question. Andy, can you unmute yourself?
Yeah.
Okay, perfect.
Okay, so I have basically two questions. One is, did you compare your subcortical structure segmentation with other software like FSL or QuickNet? The other one is, I feel like your deep network structure is a 2D. It's not based on 3D because you did that segmentation in 3 different directions and then you do a vote. So that's the questions.
Okay, so for the segmentation, yes, this is a 2D segmentation actually. And we can say it is 2.5D because we do the 2D segmentation along different direction and then we combine all of them. So this is what we call 2.5D.
Okay.
That's the first question. The second question, so in the paper, this work is actually recently published in the neural network. And in that paper, we actually compare the segmentation here with other method, which is a state-of-the-art method. I did not include it here because of the time limitation, but I can refer to that paper published in Neural Network. You can find a comparison, quantitative comparison between the segmentation for the sub-regions and other state-of-the-art method. Most of the segmentations here are quite a little bit improved by using the sub-ForkNet, we can say.
So it's better than other software?
Yes, a little bit.
Okay, thank you.
Okay, great. So Aprinda is asking a question. Aprinda, now you're unmuted. Do you want to ask the question directly?
Sure, yeah, great talk, Essam. Thank you. I'm just wondering if you've taken a look at or tested this segmentation technique in other population. I'm more interested in the aging population because deep brain structure can also tell us something about the disease population and aging population. Have you tried it in other than young adults?
Oh, that's interesting. So yeah, actually we have not tried that, but that's quite interesting. Yes, thank you. That's actually a nice question. So far we have not tried that, but it's interesting to look at.
Thank you.
Thank you.
So probably the last question is how many subjects you use for training? Fang was asking.
So the dataset we have used is actually consists of 18 subjects. So we have used a leave one out strategy, which means that we just train using 17 subjects and then test for the subject which is unknown. We have found that by doing some study, which is result is not here, it is enough to train the forknet by using from 10 to 15 subjects. So it is enough to just, if you have 10 subjects, it might be enough to have a good segmentation.
Okay. So thank you again, Essam. It was a great talk. So our next speaker would be Dennis Truong from City College of New York. Dennis, please go ahead and share your screen with us.
Okay. Let's see. Okay, can everyone see my screen?
Yes.
Well, good morning, everyone. Good evening for those of you on the eastern side of the world. I am Dennis Truong. I'm a recently minted PhD student from the City College of New York. I did my PhD there with Moran Bixen. I'm now at Soteryx Medical. So I'll preface this by just talking a little bit about applications. Our group has an extensive history of doing clinically applied models. Typically, we're looking to answer questions of locality, intensity, for whatever situation the user, the clinician is applying to. So we can do this prospectively, we can do this retroactively. One example is some of the work we did early on with pediatrics. So there was a pediatric subject that I think it was the first time an adolescent was being stimulated. So before the stimulation, we did some modeling, and we looked at the change in intensity produced between an adolescent subject versus an adult subject. That gave us a little scaling ratio. We do similar work in like animals, again, to see that intensity scaling from subject to subject, subject to species. It's also possible to do this retroactively. Sometimes a clinician will do an experiment, they'll get a result that might not entirely make sense to them, and then we'll do some modeling to try to address those questions. Example of this is early on, the first time people started using extracephalic electrodes. The MEPs that are being produced by extracephalic stimulation versus like an M1SO montage. It didn't seem to go with the current like conventional thinking that maybe moving the electrodes apart would increase current. So we modeled it, and we were able to see that there's a change in electric field induced when you move these electrodes off the head. And then, of course, models also help us visualize. So there's a lot of different ways we can do streamlines to sort of trace electric field or current density from electrode to electrode. We can look at current density or electric field projected onto the brain surface as well. This helps people intuit sort of the flow of current. It helps people both understand and then in turn develop montages for their particular application. So in brief, usually these modeling techniques, these FEM techniques have a very similar workflow. Usually we're starting off with some kind of image, we segment, we mesh, and then we solve it. In our case, we're using commercial packages. So we're using things like SPM, or we used SPM initially for the segmentation, along with some custom code developed by Andy Huang. We use Simpleware for the meshing. They have a volumetric, they have a built-in voxel-based mesher. And then we use Console for the FEM solution. Now, there's a lot of other possible open source toolboxes out there. So ROS was one. There's Fork, Comet, Synapse, Cyron. A lot of these different toolboxes, like I know ROS and Synapse, will use something like ISO to mesh, which is also a voxel-based mesher, which we should keep in mind for the next section. We apply different tissue conductivities. The main takeaway here is that usually skull and CSF are the most extreme tissues we're looking at. So like CSF in particular is very, very conductive. So that leads us to a question that we're thinking about for a while. So most of these scans, we're usually working with clinical scans that we get usually for some other purpose. If we're lucky, these will be 1 millimeter isotropic. Oftentimes, the resolution might not be as great, or even when it is, it's not always clear, especially at that interface between CSF and skull. In the case of like a T2, CSF is at least bright, so you get some contrast between the two, but when you're working with a T1, you have dark skull, you have dark CSF, and these are very, very thin regions. It's very common to get aliasing and holes that develop. So usually either through the segmentation algorithm or manually, we have to go in and then manually correct this. We go in and we correct for continuity. But in doing so, it often means that we're overestimating the CSF thickness. We're essentially filling in so that there's this 1 millimeter thin layer surrounding the brain. Now, there's different ways we could maybe address this. You could upsample scans, but that has challenges as well. So these are usually voxel-based meshers. Because they're voxel-based, if we were to double the resolution, it's really a cubic function. It would be doubling in the XYZ direction. A 2-fold increase would be an 8-fold increase in memory. And it gets restrictive as we keep upsampling. For something like the dura meninges, arachnoid pia matter, in actuality, the space that we typically segment as CSF is taken up with a little bit of this. There's proteoglycans, and there's vasculature. For example, the dura matter is probably about 1 millimeter thick in some areas. Pia, arachnoid are typically on the order of maybe 0.2, 0.1 millimeters. These are extremely thin tissues, but because they are so less conductive than CSF, if you were to factor in that volume, the actual amount of CSF is probably much thinner than what we've actually been modeling. But again, upsampling has its challenges. You could potentially do things like a surface-based meshing technique, like CAD-derived models would not, or CAD-derived like meshing wouldn't run into these problems. You could do something like partial volume calculations for CSF, which is actually something Elsa, Laxo, and Akirata did. There's other complicated, more nuanced ways to approach this. But what we were hoping to do was to essentially mimic the presence of the meninges by just a simple conductivity change. We're looking for a simple correction factor that we could apply to some of these existing toolboxes like ROS, like Synapse, that would take into account the presence of these connective tissue layers. So the plan we devised to tackle this problem consisted of a couple different levels of models. We started off with actually spherical models, just because spheres, one, these could be CAD models, they could be meshed directly from like a vector-based surface, and we can model these very thin layers much more efficiently. We could then try to emulate the effects, the presence of these very, very thin layers using just the CSF conductivity. So we essentially modeled these different layers of skin, fat, skull, dura matter, arachnoid, pia, and CSF, gray and white matter, and then combine the conductivity into one compartment, and then change this conductivity of this combined CSF layer to mimic the induced electric field created by the individual layers. We did this in a spherical model at 4 different montages. So these are spheres, it's all symmetrical. So we did based on angle. So 180-degree montage, a 90-degree montage, 45-degree, and a 4x1 configuration. We then took that sort of optimized, emulated CSF conductivity and then applied it to an image-derived model and try to compare conventional CSF with this emulated CSF to see if there was any difference. And we then took that same conductivity value for CSF and applied it to a validated data set to see if we could produce like any changes there. So the data set we ended up producing was pretty large. Probably should have graphed this in a neater way. Essentially what we calculated was the minimum electric field, maximum electric field on the surface, maximum electric field within the volume, and what we call the percent area half max. We calculated the surface area of the cortex that was at 50% of the maximum electric field and took the percentage of that surface area over the cortex. We did this for 4 different montages, and we did this for multiple conductivities just to test like a wide range of possible factors. The conductivity range ended up being fairly small. It was about 0.9 to 0.8 Siemens per meter. The midpoint was about 0.8 Siemens per meter. So we took that to be a sort of emulated value that we could potentially use to reproduce the effect of the presence of the meninges. So when applying this to a couple head models, we took 3 different heads just to get like a little bit of variability in there. We have this model of Marome that's been sort of one of our test cases. This ICBM-NY, which is an average head that Andy developed based on, I think it's the MNI like 152 or so head. And then we also have a smaller head that we use in some of our other studies as well. Typically, the subject gets the highest intensity. Typically, Marome gets a little bit of a lower intensity. So we want to get a nice little range of different subjects. We modeled M1SO, which is just a typical motor stimulation montage, as well as this 4x1 approach, and then applied both the conventional CSF conductivity as well as this emulated CSF value. And across the board, we saw this consistent increase in induced electric field on the cortex. That said, this increase was still relatively small. It was usually less than about 20% between the conventional value and the emulated value. Usually this wouldn't be a clinically significant change. I think most clinicians won't change a montage because there's a 20% difference. Typically we see that from subject to subject. to go a step further, there was some validation study that Andy published back in 2017. So to test the effect of the emulated CSF value, we tried applying that CSF value to his data set. So for those of you that are not familiar with the paper, essentially there was many subjects that were undergoing ECOG recordings awaiting surgery. These subjects had stimulation at both the frontal area and the occipital area, and these subjects had embedded electrodes that recorded their cortical electric fields. We took these electric field values, really voltage values, and compared them to modeling results. We compared the modeling results using conventional CSF values as well as emulated, and then calculated the p-values between them. We actually found a significant increase in correlation going from conventional to emulated CSF. So just to summarize everything, we found that we can actually improve the accuracy of these models by using an emulated CSF value. It's technically more accurate. Practically, it's a very small change. That said, this is a very minimal cost solution to sort of take into account the effect of the meninges, to take into account some of the aliasing that typically occurs with these very, very thin layers. So it's an approach that we suggest really most TES modeling pipelines could take. We could suggest that we can lower the CSF values from what's currently used in most places, 1.6 to about 0.85, just so they take into account some of these sort of partial volume effects. This is the citation to the paper. I'd like to thank my co-authors as well as the lab. Thank you.
Great, thank you, Dennis. It was a great talk. Okay, everyone, if you have any questions, please write it in the chat section. Another question, I don't know if Dennis may answer this or the other speakers. Is it forced to add it in all these automated softwares like PROS? Andy, are you planning to add it, or Essam, would it be easy to be added in all these segmentation processes?
So I think some of these packages, I believe you can define the conductivities yourself. But right, I think Andy or Essam could probably address that directly.
Oh yeah, right. You can use customized connectivity. But the thing is, with this paper, the thing I'm not sure is, are we gonna sell this emulated conductivity of CSF? So do we recommend this value to the research community or not?
Right, right. So I think that was the conclusion, and I think for these specific models at 1 millimeter, that's sort of our recommendation. Now, if you're doing models that are at a higher resolution, some of the aliasing effects that are sort of what we're trying to combat, that might not be present. So I don't know if I would use it universally for something like SES stimulation or DBS or different paradigms like that.
What about the non-invasive ones, such as tDCS or tDCS? It would be helpful to add, right?
Right, right. So I think most of the models that we work with that are, 1 millimeter isotropic scans that we're segmenting at that level, we suggest using a lower CSF connectivity just to compensate for some of these effects.
Okay, great. Thank you again, Dennis. I'm going to clap on behalf of everyone here. I'm sure no one can unmute themselves. Okay, in the interest of time, we are going to invite our next speaker, Jose Gomez-Tames. Jose is from Nagoya Institute of Technology. Good night, Jose. So please share your screen with us.
Okay. Good night. Thank you very much for the invitation. Okay, I'm sharing my screen. Is everything okay?
Yes, you're all set.
Okay, well, thank you. So yeah, I would like to talk about one of our recent papers, significant group level hotspots found in deep brain regions during transcranial direct current stimulation. So first, one of the points that we are investigating in this paper is about the different neuromodulation effects. Basically, there is, we can divide in three different levels. We can use this classification. One is the tDCS neuromodulation effects that are direct. This is direct considering that the tissues that are beneath the electrodes, they have the stronger current intensity or the electric field. So basically, we can consider that we will have we are confident that we have a direct neuromodulation on these superficial brain regions. The second one is that we have may have a side effect, if we want to say it like this, that is an indirect neuromodulation of other regions like deep brain regions through associated cortical networks. And the one that we are more interested in this work, in this presentation, is the potential direct modulation also of deep brain regions. Because in the case of tDCS, where we have a large electrodes and the electrodes are far away from each other, this is a bipolar configuration, we may have a spread of the current. So this spread of the current, this dispersion, may allow to reach deep brain regions with maybe significant or high values. So we want to see this link between the how is this possible or not. So there is actually evidence, a lot of evidence on this. For instance, we have seen from fMRI studies, we have seen the activation of deep brain regions from tDCS. Also, we have experimental measurements where we can see also values, electric field values, significant, also in deep brain parts. And finally, this approach that I will discuss more of this during this presentation is using computational modeling. So using computational modeling is also possible to predict hotspots or high fields on deep brain regions. So despite all of this evidence, still mostly these direct effects on deep brain regions are usually not mentioned, or at least not considered very strongly. Maybe one reason is that there is a perception that maybe this tDCS delivers both to the cortical and deep regions. However, mostly the most consistent neuromodulation will be in cortical regions, in superficial regions. And this is basically because when the current is injected to the brain, we have, as I said before, we have a good confidence that we will have high values near the electrode. But after the dispersion and due to the intervariability in subjects, the dispersion of the current can go to different places. These effects might be very variable between subjects and among the subjects. So to investigate this point, we use a group level analysis. So here's a summary of the two approaches of the computational tDCS to investigate electric fields. The first one is that we use in the upper part, as you can see, we use individualized head models. So we have a montage. We know the properties of the electrode. We know the positions that we want to investigate. And we can observe the field distribution. So this is very important because we can have a very detailed information of effects for one subject. However, if we want to generalize or investigate from one subject these effects to a population, there might be some bias. In the second point is that we use a group level analysis. So it's the same. We apply a fixed montage to a population. So we want to see different electric fields for all the subjects. So after this, we can investigate intervariability between the subjects, and after that we can also investigate what is the group level effects. So for that we can use some techniques, some registration techniques, in which we can have an average, or we can investigate the standard deviation to see what is the general effects, to a more generalized to investigate the effects on the population. So for this study, we will go to this second approach, that is the group level investigation. So basically, the aim of this work was to evaluate the electric field in deep brain regions at group level using one-for-all montage. And for that, we use high-resolution computational models with registration techniques. And we use a post-processing analysis at group level effects. This is the publication. So if you want to see in more details later, the points that we are discussing now. Also, there is a very interesting editorial that was also published in the same issue, where there is different directions and different very interesting opinions about this has been. So I recommend also reading this editorial. So for the methodology, we use head models using the semi-automatic segmentation method that Essam explained before. Also, we are one advantage of now the work from Essam, that by using the deep neural network, we can have head models ready for a large number of people. So we can apply these techniques to populations of now, for example, adults, or maybe in the future, we can also investigate effects for aging populations and etc. This is some work that is interesting, and we are looking forward to continue working on this. So for that, we use these registration methods techniques. So very simple, just let me mention that we need to apply some transformations in order to project these fields into a template. So since we have the information in a template, we can basically have a averaging or apply different statistical methods to investigate the effects on a group level analysis. And here is the segmentation for the deep brain regions that we are under investigation here. Okay. So I would like to go into the details of the results. And the first point that I would like to show to you is the interindividual difference and registration effects. Here I'm showing the deep brain regions, the electric field on 18 different individual head models. We can observe that we have certainly we have a variability. So for one subject, as you can see here, the upper part, the electric field is very low, but for other subjects, the electric field is very high. So even when we are using this one fixed or one for all montage, we will have high variabilities between the different subjects. And here it can be quantified by using the standard deviation. So we can find that there is a standard deviation normalized up to 40%, in some other parts of the deep brain regions up to 20%. So also the electric field, the peak values can be changed up to 40%, 14%. So, well, we can find that there is a large variability. So as I said before, by using a personalized head models for one subject, maybe the implications of one subject may be not generalized for the population. So it's important to consider this group level analysis when we are using the one for all montage style studies. So this is just to show this point. The second result that I want to show is the actual group levels electric fields in the different in the brain cortex and the different regions for different montage. So the main result here in this slide is that it's possible to have consistent group level hotspots. These are robust across the individuals. So for instance, in the C3FP2 montage, we can see some hotspots in the caudate and also in the putamen. For instance, in the FPCOZ, we have a significant consistent hotspots on the brainstem. So depending on the selection of the montage, we can try to target specific deep regions where we can deliver consistent hotspots for a group of people. So this is the result that I want to show here. In the next slide, I would like to show that this is not only consistent, but also the values in the different regions are close to the cortical values. So this is the normalized electric field for the normalizing with the maximum value in the brain cortex. So for the F3, F4, we can see that the electric field on the caudate is around 70%. So this is a very significant value. For other montage, for instance, the CCOZ, the electric field in the deep brain regions, they do not reach over 40%. So here we have low values. So basically, for the different montages that we use, we found that the electric field intensity is around between 35% to 70%. And 70%, we have the C3FP2, F3F4, and C3C4. And these are not only the peak values. I mean, this is not only for very specific points on the surface of the deep regions. Also, we have a large relative volume of different parts of the different regions are also have significant values. For instance, F3, F4, for the caudate, 50% of the volume has an electric field over 0.5 volts per meter. Here we are using 2 milliamperes of stimulation for the injection current for tDCS. Okay. Then what about the other montage? Here we are only using the large bipolar or distant electrodes, so to make the high dispersion, so basically we can reach deep brain regions with a high current. By using a large electrode, for example, 5 by 5, or by using only 5 centimeters square, for both cases, even large or small electrodes for a bipolar montage, we have a similar distribution. We can reach values close to 70%. But if we use a small electrode or focal montage, the values in the different regions are lower than 30%. So another way we can say that we can avoid directly engage in different regions by using this small montage, small electrodes, or using this high definition montage. This is one option for this. Okay, so now I just would like to have here one parenthesis. So what happens, for instance, for other kind of stimulations like a transcranial magnetic stimulation? Here we also have a publication and we address this question. So I just would like to very briefly mention what we found here. So, well, of course, the tDCS and TMS, the applications are different and physical mechanisms are not the same. So in TMS, with the electric field decays very fast with depth. And we have a physical limitation in the trade-off between depth and spread, which is different from the tDCS. In this paper, what we did is to optimize the coil position for group level. So we found what is the optimal coil position to reach the maximum electric field on different regions. So even by using the optimization of the coil for a population, we could not have electric fields higher than 50% on the different regions. So as a comparison, just the tDCS was possible to reach 70%, and for optimized TMS coil positions, we were not able to reach more than 50%. So I think this is maybe an interesting comparison. So basically what this means is that if we want to reach higher fields on the different regions, we will need to stimulate with higher electric fields the brain cortex. So of course we will have more side effects, and maybe the focality will not be the same for the case of TMS. Okay, so I just like to conclude. Basically, well, the tDCS mechanisms at deeper regions may include not only indirect modulation via underlying cortical or subcortical circuits, but also direct modulation of different regions. And this conclusion we find by two ways. We found that there was consistent deep hotspots across a population for one-for-all montage. And also we found that the electric field intensity in these hotspots were comparable to the cortical regions values. So one of the implications of this work is that, well, we can use MRI-based individualized head models to try to target the specific brain regions. However, maybe this is not available for every trial, maybe possibly in the clinical practice. So this group level analysis can be used to optimize one-for-all montage for a population without MRI-based individualized head models. So this may reduce time, and may reduce also equipment allocation for large trials. And something that I think is very interesting is that many past and ongoing trials with tDCS may have been already consistent activating or modulating deep brain regions. And maybe this has been ignored or has not been mentioned in some of these studies. So it's important that when we are using these large bipolar electrodes, we should be also consider that we are not only may have a direct modulation on the brain cortex, but at the same time, we may have also some effects on different regions that may also affect the conclusions or the different circuits we are trying to engage. So if we want to avoid this, we may try to use small electrodes, or if we want to engage deep regions, we can consciously use large electrodes. There are also other techniques like for instance, interferential currents. That maybe we will discuss this in the next presentation. That we may also possible to reach deep regions modulation. So, well, I would like to say thank you again to all the collaborators, to the group in Nagoya Institute of Technology, and thank you very much for your kind attention.
Great, thank you, Jose. It was a wonderful talk, and I'm going to clap on behalf of everyone. Aprindo, I'm going to unmute you. You can ask your question directly.
Yeah, thank you, Zeinab. Thank you, Jose. Great talk. I was just wondering which registration technique did you use for your images, and whether you use any template to register all the images to be in a standard space.
Yeah, exactly. We use the standard space, maybe ICBN 152, if I'm not wrong with the name. So yeah, so basically we use this, maybe the free software also allows some of these registrations. For the case of the deep brain regions, we use the minimum distance from the register values to the template. This has maybe around 5% of error in average.
Is that a linear or non-linear registration technique?
Yeah, in this case, it's linear, yes.
Okay, thank you.
Thank you.
Thank you. Gaby was asking question, it's resolved, or, I mean, you can unmute yourself if you want to. Okay, so Andy is asking, Andy, now you can unmute yourself, ask your question directly.
Yes. So I just want to ask you, did you optimize your electromontage in your simulations to get all these hotspots in the deep brain?
Yeah, thank you. Yeah, for the case of the tDCS, we only use the, as you saw, the common EEG system. So we use the common montage, and we apply this for all the subjects. So they were fixed montage, and they were not optimized for the patients. They were applied like we will use in a clinical trial. So even though that the electrodes were not optimized for each subject, it was possible to find these consistent hotspots.
Okay, thank you.
Okay, another question. I don't know if probably you answered it at some point. I missed it. Is it sensitive to the conductivity that you assign to CSF?
Yeah, this is a very interesting point. For the CSF, I think maybe for CSF, we are quite confident on the values. Maybe the variability to CSF is not so large as for instance, for the other brain tissues, like the gray matter, for example. So for CSF, we do not expect so much effects, I think. For the other tissues, of course, there is a sensitivity analysis. There could be some variations. But I think that conclusions, there will not be affected. The main conclusions about the consistency of the hotspots will be even if we apply a different set of conductivities, will be the same, I believe.
Okay, great. Ghazale is asking, first she says, great talk. Why did you focus on peak electric field? Did you use maximum electric field or percentiles? And have you tested mean or medians as well?
For the electric fields, yeah, we use the, well, for instance, in this figure, this is the average, no, I'm sorry. For these figures, this is the average of the electric field, the spatial average of the electric field. For these ones that we are showing here, this is the peak values on the, for each deep brain part. So also also we tried, I think, the mean values for the deep brain regions as well. And also we tried the volumes of the deep brain regions. So for a certain threshold, what will be the volume activated within a certain deep brain region? So by using these different kind of metrics, we were able to have consistent conclusions that I was discussing here.
Okay, another, probably that's going to be the last question. How mesh quality will impact the electric field distribution in deep brain regions?
Can you repeat again? I'm sorry.
How mesh quality will impact the electric field distribution in the deep brain regions? So technically, how mesh quality matters.
Mesh quality.
Mesh quality.
Mesh quality. Yeah, sorry. Yeah. So yes, so maybe I did not mention that in too much detail. The technique that we are using is called scalar potential finite difference. So for this technique, the mesh quality, basically these are voxels, and the mesh quality for this kind of techniques is basically how finite is the voxel size. For this model, we were using 0.5 millimeters. We also made tests using half that value, 0.25, and we did not find a significant difference. Maybe using over 1 millimeter, we may have some effects, some variations. But when we go below 0.5, or of course, if we use 0.25, the variations are small and the results are consistent.
Okay, great. Thank you so much again, Jose.
Thank you very much.
Thanks. Okay, so perfect. Let me share my screen. Okay, can you see my screen? Perfect. What I'd like to talk about today, first of all, I'm Zeinab Esmaeilpour from City College of New York, and happy to have all of you here. And the topic that I'd like to talk about today is temporal interferential stimulation and how it can target deep brain regions and also how it can modulate neuronal oscillations. Just a quick recap. Temporal interferential stimulation or temporal interference stimulation is a method that technically delivers two sources of high frequency stimulation over the scalp, which produces two major waveforms throughout the brain. In the cortical regions, we would get unmodulated high frequency sinusoidals. And in the area that these two sources intersect each other with the same intensity, they would produce amplitude modulated waveforms. And with using the modeling method, we can see, of course, this method would produce a strong amplitude modulated waveforms in the deep brain regions. However, it will produce really a strong or maximum electric field on the cortical regions, but with a different waveform. So cortical region would be exposed to purely unmodulated high frequency stimulation or minimally modulated. However, in the deep brain region, they would have modulated waveforms. But the whole method is based on this hypothesis that neuronal membranes has this intrinsic characteristics, which is called low pass filtering property, which does not let the neuronal activity to follow the high frequency components of the stimulation. So it leaves the cortical areas untouched. However, in the deep brain regions where we produce amplitude modulation, neurons would be responsive to that. And also we have Grossman's study, which shows exactly that neurons are responsive to amplitude modulated waveforms. However, they are not responsive to purely sinusoidal unmodulated waveforms. But the question that immediately comes to mind is, wait a second, even the amplitude modulated waveform does not have a frequency component in the low frequency. And the frequency component of the amplitude modulated is in the kilohertz range. So how is that the central nervous system is sensitive to this waveform, which has obviously high frequency component, but it's not to unmodulated ones? So the two main questions that we started to ask throughout our study was what makes central nervous system more sensitive or more sensitive to amplitude modulated. However, it's not that sensitive to unmodulated ones. The matter of selectivity, how central nervous system can select to be sensitive to modulated one, but not to unmodulated ones. And also knowing that the amplitude modulation has a high frequency component in it, how much intensity do we need to use in the scalp in order to produce the electric field that can modulate significantly oscillations in the deep brain regions, and which brings the idea of sensitivity, how sensitive the network is and how selective it can be. In order to answer this question, we started using pharmacologically induced gamma oscillation, which has been shown previously that is highly sensitive to electrical stimulation. So here you can see these experiments are in vitro slices of rat hippocampus, which are washed by carbachol, which is a cholinergic agonist. And if you record from CA3 regions, we are recording from CA3A. But in the CA3 regions, we would be producing gamma oscillations, which is really stable for over 2 or 3 hours. And the electric field that we applied here was in a direction that would make the pyramidal neurons in the CA3C regions depolarized. So we use this system to systematically contrast the effect of low frequency, high frequency sinusoidals, and also contrast it to amplitude modulated waveform with the same carrier frequency in order to see the difference in sensitivity and selectivity to these waveforms. And in order to explain the observations, we use the network model, which has been developed before, a network model of single compartment neurons of adaptive exponentials, which has the excitatory and inhibitory neurons, which we can talk about it later. Okay, the experiment was, all the experiments here that we are going to talk about are the acute experiments. So all the slices are subject to 2 seconds of a stimulation. We have 1.5 seconds of baseline, then 2 seconds of a stimulation. And after that, we have 1.5 seconds of after effects. And the interesting thing is we see 3 different profiles of response. In the low frequency, which is 5 Hertz here, you can see that ongoing gamma oscillation is modulated by the amplitude, by the sinusoidal waveform. And I was able to resolve the effect for even lower than 5 volt per meter, but now the effect is, the spectrogram that I'm showing here is for 5 volt per meter. Then we have another profile for amplitude modulated waveform. And as it is obvious here, ongoing gamma oscillation is modulated by the envelope of amplitude modulated. So the stimulation here is 1 kilohertz carrier frequency with 5 hertz of envelope. And also I use 80 volt per meter as a field intensity. So we can see in order to produce the roughly same effect, we need to increase the field intensity in order to produce the significant effect. And also another profile of response that we see in these experiments are the response to unmodulated kilohertz stimulations. In here in the spectrogram, I'm showing the response to 2 kilohertz, which there is the onset response at the beginning, and then it goes to the steady increase of the gamma oscillations. Okay, so for each of these experiments, each of these conditions, I use different intensity in order to see the dose response relationship. So as we see here for the 5 hertz low frequency, we can see that from 1 volt per meter to 5, gamma oscillation is modulated by the electric field, and also the effect is symmetric. For the depolarizing phase, the gamma oscillation is, the power of gamma oscillation is enhanced, and the depolarizing phase is suppressed. For the purely unmodulated high frequency stimulation, for 100 hertz and 2 kilohertz I'm showing here, I use 4 different intensities for each of them. And we can see that at the beginning, there is this increase in the response, and then it decays, and then it stabilizes for the second half of the stimulation period. We only observed the increase in the positive phase of the stimulation, which probably reflects the sensitivity of the active network to the depolarizing phase of the stimulation rather than the hyperpolarizing phase of it. Then another important thing that I want to talk about here is the intensity that we use. With increasing the carrier frequency, we have increased in the response. Okay. So for the amplitude modulation waveforms, we see the same thing. So for all the amplitude modulation waveforms that we have here, carrier frequency is fixed. So we have all of them have 5 hertz carrier frequency, but the envelope is changing from 100 hertz, 1 kilohertz to 2 kilohertz. So for each of these, we can see that the gamma power is modulated by the amplitude modulated waveform. Obviously, with increasing the frequency of the carrier, we need more intensity in order to produce the same response. In other words, the sensitivity of the network is going to decrease with increasing the frequency of the carrier. And in order to quantify our responses here, we introduced 2 different measures, dynamic modulation and static modulation. Technically, dynamic modulation captures the fluctuations of the power in the amplitude modulation, which is like the ratio of the peak to the trough. In order to not to include any artifact, we started quantifying that for the second half of the simulation. So all the results are from the time 2.5 to 3.5. And then there is 2 different things that I want to discuss here. First of all, we expect for the amplitude modulation stimulation that for the peak of the envelope, we would see the enhance in the gamma power. But then when we have this 0 electric field, we would expect to be on the baseline. But as we see here in all of these waveforms, if we go with the gamma power doesn't go back to the baseline, it suppresses, as we see that the same thing when the stimulation is terminated. This calls for some homeostatic adaptation mechanisms that we are going to talk about it later. And what we see here is the dynamic modulation technically increases the electric field that we can see here. With increasing the electric field in each of these waveforms, we would see the increase in dynamic modulation, which is monotonic. And also increasing the carrier frequency, it technically drops the sensitivity of the network, which could be predicted by the low pass frequency properties of the membrane. And also for each of them, this is the static modulation that we see here. And we can see that on average for each of these waveforms, we have the average enhancement of the gamma power for both modulated and unmodulated ones, because even though we have the suppression here, which we are going to talk about it later, this suppression is not symmetric. Okay, now with all of these things that we have seen, we have started to use our model to explain the effects that we see here. As we see, our model is perfectly following the experiments, but we learned two important, we had to incorporate two important things, and we learned two important lessons from using these models. First of all was that in order to reproduce the effect that we are seeing in the experiments, we should decrease the time constant of the neuronal membrane that we have in the model. So all of these models that we have here in the network are, excuse me, are single compartment. So that single compartment technically represent the most sensitive compartment in the model, in the neuron. So usually in the active networks, the time constant of a neuron is 3 to 10 milliseconds. Here we had to change it to 1 millisecond in order to match the results that we are seeing in the experiments. And that calls for some thoughts about probably there are some other compartments involved in the network that are more sensitive to this kind of waveforms. For example, it could be the axons that are faster and have the lower time constant. So this was one of the take-ons from the model. The other one was, so the time constant would technically talk about the sensitivity, but we have another issue of selectivity. This network is more responsive to amplitude modulated than the unmodulated ones. As we saw, even for the high frequency stimulations, the pure unmodulated ones, you have this increase and then it drops to a stable one. So there is this homeostatic adaptation mechanism that is bringing the system back to the baseline. That's why in the amplitude modulation on the time that we don't have any electric field, the power of gamma doesn't go back to baseline, but it suppresses. Even at the end of the simulation, and when the simulation is terminated, the same thing happens. So we incorporated the GABA B as a homeostatic mechanism that has higher threshold, that whenever the system is getting super active, with a delay, it start activating and suppress the whole system to bring it back to normal. And this is technically on the same topic that I talked about, just for a bit more detail to show the issue of selectivity, that using amplitude modulation in any system, the current that we use and how selective the system is to amplitude modulation versus unmodulated ones depends on all their homeostatic mechanisms, or like the regulatory mechanism that they have, and also about the time constant of the whole network. And as you can see here, I'm sure it's so complicated, let's walk you through this. This solid blue line is the one that in both of these reproduce our results in the experiment. So let's imagine we don't have GABA. If we remove GABA, the selectivity to dynamic would drop, and also for a static we would have higher response. So this emphasizes the role of GABA B in the selectivity of the network to amplitude modulation rather than unmodulated ones. And also for the time constant, increasing the time constant from 1 millisecond to 0.4 would increase the responses significantly, and also reducing it would kind of disappear. Okay. And also another thing that I really love to dig more and also talk about it is the spiking activity. In the model, I try to look at the spiking activity during the simulation. For the models, we had the significant effects for 40 or 80 volt per meter, but for 20 we didn't. However, in the spiking activity, we see some random increase in the spiking activity, but which is not enough to modulate the gamma oscillation. In order to see spatially, if we have the interferential stimulation in the brain, how would all this modulation spatially distribute over the head? We technically use each voxels in the brain, and we assume all of these voxels have the same sensitivity as our network, which we know this is an assumption, which is not true, and then try to spatially see how these modulation would look like. So this is the spatial distribution of the dynamic and static modulations. So we can see the sensitivity is a function of time constant, and with decreasing the membrane time constant in network, technically we need less current to produce a significant enhancement in the gamma oscillation in the deep brain regions. And also in these ones, it is obvious that removing the GABA B, which remove the selectivity that we see, so the system is not more sensitive to amplitude modulation versus unmodulated ones. See, we have a lot of static modulation in the cortical areas, which it's not the, it would happen, but having the GABA B would, or any kind of homeostatic adaptation mechanisms in a network, that would make the system more selective to amplitude modulation waveform rather than unmodulated ones. And yes, so in conclusion, we tried to model the amplitude modulation, and we saw that cortical areas are exposed to unmodulated waveforms with high intensity. However, deep brain regions are exposed to amplitude modulated with less current or less electric field. And also the sensitivity, meaning how much current do you need to use on the scalp in order to produce a significantly modulated oscillation in the deep brain region, depends on the network time constant. So if network is in highly conductive state, it would need less of a current to produce the same effect. And also selectivity is a function of adaptation mechanism, at least in a network that we were studying, and that makes the system more sensitive to amplitude modulation, not unmodulated waveforms. Okay, thank you everyone. Let me stop sharing my screen. Okay, let me, Dr. Pirachev, I'm going to unmute you if you want to ask your question.
Yes, thank you for this opportunity.
Yes,
very
sure.
interesting presentation.
Thank you.
My one of the main questions I have had about interferential stimulation is whether it can be tolerably delivered in humans transcranially without anesthesia, right? We know that, of course, with anesthesia, you can do ECT, which has very strong fields, and that's okay. But we know that with TES, we usually are talking about a few milliamps of current that can be, at most, that can be delivered tolerably. So I'm wondering, when you're talking about in simulations about currents that are on the order of 100 or 200 milliamps, and I understand these are high frequencies, but still these are frequencies that we use for TMS in pulsed form, a few kilohertz, and there's definitely this scalp sensation. So I'm wondering if anybody has either tried experimentally, or if there is any simulation or other argument that this is feasible from that standpoint, from the standpoint of tolerability.
For producing 80 volt per meter, definitely we need really high intensity, and I'm sure it's not tolerable. We have tested in our lab like 20 volt per meter with like 1 kilohertz. It's not that pleasant at all, and I'm sure more than that it would be really painful. And but the thing is, we have to. These are our educated guess for the system that we are testing. So we don't know if we take all these things to human, the human brain would be more sensitive to these kind of waveforms or not. That yet need to be some experiments to be done. I know some groups are doing some experiment, but not using really high intensity. But if we assume that all the predictions here are correct, then using 100 volt per meter, 100 milliamps, technically it's not feasible and it would be super painful. At least that is my understanding. Okay. Here, I'm going to unmute you if you can ask your question.
Sure. I mean, I am relatively new. I do a lot of aphasia recovery, particularly in stroke. So, and we're using TES, tDCS, and different techniques. So, I just wonder. I mean, I see the amplitude modulation. So, hypothetically, if one were to use frequency or phase modulation, how would that interference? I don't know, is it clinically applicable? Has anyone simulated? And if one were to use different waveform for the high frequency carrier, a different waveform, I don't know.
What do you mean by different waveforms? Like not sinusoid or
yeah.
I mean, pulse?
Yeah.
There is, I think the whole method is based on having the sinusoid that we can produce amplitude modulation. I don't know if you have pulse waveforms. I'm sure, I mean, my guess is the waveform that in their intersection would be produced, it has different frequency components, if I'm not wrong. So no, to my understanding, amplitude modulation is by definition using sinusoidal waveforms. And it has been used before in the peripheral nerve stimulation, muscle, and now it's been introduced to brain, but none of which that I know of have used pulse. I have seen bursts modulated waveforms that they can be pulse, but not the amplitude modulated ones.
Cool, thank you.
Great. Bosho Wang, I'm going to unmute you if you can ask your question. I think I unmuted you.
Hi, this is Bosho.
Yeah.
My question is that in an in vitro experiment, you had 2 electrode, basically wires, parallel wires to replicate the electric field, like uniform field, and to generate like an amplitude modulated field. Have you ever tried to use 2 pairs of those electrodes to generate actual temporal interference field, which is different because it has unique spatial temporal features? And so in this case, although your models and your experiments are consistent, they are not TIS.
If I get your question right, you're asking, instead of having these two sources that can produce some amplitude modulation waveforms somewhere in the brain, we use just two pairs of electrodes which can deliver amplitude modulation on its own, right?
You have one pair of electrodes that generated one electric field across the brain slice. Have you tried to use two pairs of electrodes to
No,
generate?
no, we didn't. No, it was so complicated. So the whole experiment was based on that. We are recording from hippocampus, and we imagine that hippocampus has been targeted by temporal interferential stimulation. So if we produce the same electric field that it would see in that situation, which is amplitude modulation, how would be the response?
The point is that the hippocampus is deep in the brain. So if you imagine that the electrodes on the surface generated the E field are not aligned in the brain, in the deep structures. So that in that way, using one pair of electrode would not be able to replicate the temporally interfering E field. So that's a question.
Yeah, if that is a question, we didn't answer that. We assumed that in the direction that we are testing in the experiment, there is exposed to the amplitude modulation waveform, and we asked this question of how it would respond. But yeah, if you're talking about,
would it be difficult to set up two pairs of electrode for that experiment setup?
I think it would not difficult, but I don't see it's worth all the hassle. You need technically two carrier source generators, and you have to, I think the setups are too small, so fitting two different wires in that setup would be difficult, given the space limitation that we have. And also you have constraint on how to place them. So I don't think it's worth trying. But
okay, but think about it again.
Yeah. Thank you for the suggestion. Okay. Yeah, Dennis is asking, high frequency stimulation is a big open question in SCS too. Yes, it is. Recently, not recently, probably like in the last 10 years, spinal cord stimulation is using high frequency stimulation, such as 1 and 10 kilohertz. And Dr. Peter Chaff, I guess we answered that question, right? I don't think that would be tolerable, having 100 or 200 milliamps on the scalp without anesthesia. So that would not be possible. I mean, we tested in our lab. I cannot even tolerate the, yeah, obviously having a higher carrier frequency would makes it more tolerable. So I would say I've tested 1 kilohertz on my hand with 10 milliamp. It was kind of okay, but with 20 it wasn't. If you increase the carrier frequency, of course, it would be more tolerable, but at the same time you are losing the sensitivity. So in my idea, I don't think 100 or 200 milliamp would be tolerable using 1 or 2 kilohertz carrier frequency. Have you investigated selectivity and sensitivity targeting in the cortex? No, but it has been done before. So I probably refer you to the Grossman's paper. They show the whole promise of temporal interferential stimulation is that you can steer the target of the targeting a deep brain region that is exposed to amplitude modulation waveform in any location that you want, depending on how you optimize the location of the electrodes over the scalp. So that the study has extensively investigated this one. Okay, so it seems that's it. I'd like to thank all the organizers that did such an amazing job. And also I want to take a moment, thank all the speakers who talked in this session.